A very good evening to all the viewers of today's webinar. I, Dr. Anand Chintakrindi, Assistant Professor, Vivekanand Education Society's College of Pharmacy, welcome you all on day 2 of this webinar on Design of Experiments in Pharmaceutical Research, a primer for academia. Let me take this opportunity to thank all the viewers for their overwhelming response on day 1. Our yesterday's session on YouTube has received more than 1200 views, in which our resource person, Dr. Anilkumar Gandhi, started with the basics of statistics, and then he enlightened us with certain DOE, that is design of experiments terminologies, which was followed with the comparison of OFAT versus DOE, that is one factor at a time versus design of experiments. Then he elaborated the process of DOE, and finally he tipped off with a brief discussion on factorial design. Let us look what does he have in store for us today. Over to you, Dr. Gandhi, sir.
Hi, thanks, Anand sir, for a nice background and the information that he shared about summarizing yesterday's event and yesterday's session. So, without any further ado, let's get started with the session today. So, today we are having the day 2 of design of experiments, and as you will appreciate, the more we get deeper into the statistical part and the concept part, will go more and more in depth. However, I'll try to make them easier for all of us to understand. I think this was the same disclaimer. I've just put it again, so we'll skip this. Like we did in the QBD webinar, what I have done today again is that I've picked up some of the responses where I found that there were some challenges. So we'll just revise this concepts and we'll relook at it. Like the first question that I picked up was, which of the following is true with respect to OFAT? That's one factor at a time. And then I had given 4 options. What I saw here is 50% of people, students have answered it, that it often leads to a lot of experimentation, which is perfectly fine. Yes, that is the answer. However, few others have given like 21.9 or close to 22% people have given it provides factor interactions. So I just want to clarify that the reason why we don't use OFAT is because it does not provide factor interactions. And what do we mean by factor interactions? We'll see it today. I'll be explaining you in detail, so hopefully it will become more clearer when what do we mean by factor interaction. This next question was an important step after determining which factors have a significant effect on the response to Pareto chart is. So what I really meant was, if you all remember that I'd given a tip, that was a slide with a tip written there, which is, it said that before we go further, we must check or we must confirm our results through ANOVA. So the right answer, of course, is the ANOVA, which is like about 60% of you have got correct. But there are few people who have got equal portion of the other answers. That is, go for confirmation run to confirm the observations of Pareto. Please remember, the confirmation runs come only towards end. If you remember that ANOVA gives you an equation And also you will see in the optimization that we'll do tomorrow is it also gives you the optimum level. So with that optimum level, you will put it in the model and you will of course see what is the predicted response, but you will also do or carry out experiments and you will see what are the observed responses. And then you will compare the observed responses versus predicted. So that is the last step and does not come immediately after Pareto. There was another option which is experimentation is over as significant effects are determined. No, I think experimentation does not get over at half normal plot or Pareto chart. See, what happens is that half normal plot and Pareto charts are very kind of a visual aids because it shows you very clearly what are the effects. And therefore, it is an aid which assumes certain things and we must confirm it through ANOVA table. So the right answer is that after you have seen half normal plot, after you have seen the Pareto chart, you must go for ANOVA table. Then the next was half normal plot. Now here, the options, there were two answers to these options. And that's where I see 30% have got it, rather I should say 14% and 46% of this have got them correct. Now let's go to first the chart again and then we'll go back to the options. When you see half normal plots, as you see here, the effects which are away from the trend line are the ones which are significant effects. So the factor C, which is PVP concentration here, factor B, which is cross-Karmelos concentration, have significant impact on impurities, whereas rest all the factors, including the factor A, which is MCC, that combination of factors, they have no effect, that is insignificant effect. So the first answer, of course, is that the significant effects are located away from trend line. That's the first answer. The significant effects are on the trend line. That is not right because on the trend line, what you have is the insignificant effects. So if you had corrected, you have put answer as insignificant effects are near trend line, that is also right answer. So you can choose either the blue one or the orange option. So those are the ones who got it correct. You certainly do not have significant effects on trend line, neither insignificant effects which are away from trend lines. I hope this is very clear because this is the first step of analyzing the data. Once you fill your response in the factorial design in any of the software, what you see next when you go for analysis is the Pareto, half normal plot and Pareto, and that's the reason I'm taking this once again. In Pareto, if you see, the question was, excuse me, any effects represented by a bar line in the Pareto chart, which is below T value, are considered significant effect. Now, actually, for this, majority have got wrong because this is false. This is the T value. You can see T value limit here is 2.57. Any bars which are less than or which are below this T value are actually insignificant effect. And if you see in Pareto chart, the insignificant effects are filled with the solid colors, whereas the significant ones are having a border kind of thing and the white inside, which is a kind of hollow bars. This is also one way to see it differentiated. But most important thing is, first thing we see is the Bonferroni limit. Anything above Bonferroni certainly is the critical or significant impact factor. Anything which is below T value is surely insignificant. Anything in between is what you will see the ANOVA table and confirm, which is the borderline case. I hope this is clear. And with this, I think we'll get started with today's session. And in today's session, I want to start with an interesting concept of type 1 and type 2 error. I want you to a little bit focus on these terms because it's little confusing terms, and therefore let's get into what do we mean by type 1 and type 2 error. So type 1 error is caused when hypothesis that should have been accepted is rejected. What it means is that actually I have a positive effect. Let's say we will see with example where it will get more clear. But if I have a PVP which I know that it has a effect on dissolution, but for some reasons after I generate data and I do the analysis, what I see is PVP effect is insignificant, what it means is that I have got a wrong result. It should have been acceptance. I have in fact rejected it, right? So that's the type 1 error. It is typically represented by alpha, and it is called as a label of significance. Now, there is a very important aspect associated with this type 1 and type 2 error, and that's why we are discussing it. Yesterday, we saw that in ANOVA table, any effect where you had a p-value less than 0.05, we would consider that as a significant effect, and any effect where it is more than zero point 0.1, just to be on the safer side, anything which is beyond 0.1, you will say it is insignificant. So that 0.5 actually comes, sorry, 0.05 actually comes from type 1 error. And what we are saying is that when we accept at the level of significance of 0.05 means that we are 95% sure. There is always 5% probability that we might be wrong, but I'm 95% sure that when I say that this particular factor has an effect, then it does have an effect. So that kind of confidence comes when you put that significance value as 0.05. So that's the type 1 error. I'm sure that it might be a little confusing now. It will be clear with the two examples that I'll give you in the subsequent slide. Now, what is type 2 error? Type 2 error is an error that occurs when the null hypothesis is accepted when it is not true. So you are actually supposed to have a negative effect, which means, let's say you have an MCC, which is not going to impact your dissolution, but you say that MCC does impact the dissolution. Now, why that happens? That happens because of type 2 error. Now, what is the type 2 error called? It's a false negative result, and it is denoted by beta. So alpha is type 1, beta is type 2. And very important thing is that type 2, like type 1, relates to the level of significance or 0.5, 0.05 value in the ANOVA table. The beta level or the type 2 error mainly responsible for the power of the statistical test. What do we mean by power? Power means your ability to differentiate between the noise and your data that is generated because of your factors, in fact. Okay, so let's see this with an example. So this is a very striking and little funny example. So you have two people. One is a pregnant lady and a male person, and both of them, you see that they undergo pregnancy tests, let's say. So what happens? Obviously, you know that the lady is pregnant, and you know that men cannot get pregnant. So your actual value is, as he has shown as 1 and 0 in this slide. But suppose you have used a pregnancy kit or a testing kit which shows that the lady is pregnant. Perfect. That's what it should be. But it also shows that the man is also pregnant, which is not the case. So the false positive results, what you get, this is called as type 1 error. And when you look at the second part of it, now the pregnancy test says that both are not pregnant. Now there is another problem here, because you know that the lady is pregnant. So now what you've got is you've got a false negative. You know lady is pregnant, but the test is showing that she is not pregnant. And of course, man is not pregnant, he cannot get pregnant. So it shows correct results, which is true negative. I hope with this example, the concept of type 1 and type 2 error is quite clear. Type 1 is the false positive, type 2 is the false negative. Let's see it with our paracetamol example, paracetamol IR tablet studies, what we have taken. Now, we know that what is the reality? PVP concentration impacts dissolution. We also know that MCC concentration does not impact dissolution, or MCC type does not impact the dissolution. Now, what is the outcome? Now, outcome is, it says yes, impacts the dissolution. So when we look at the PVP part, perfect. That's what we know it should be technically, and in reality, PVP does impact the dissolution. So it's a good decision. But you get yes also for MCC. If you do get yes for MCC, then what it is said that you've committed a type 1 error. Like we said that men also has become pregnant. So it's similar analogy like that. Now, if you take second outcome of the result is the no, that means the factor does not impact dissolution. Now, if you say for the same negative result for PVP, then you are committing a beta error or type 2 error, which is the false negative, because you know that PVP is supposed to impact dissolution, but your data or your analysis says no. So that's the beta error. But if you say the MCC does not impact, that's the good decision. So again, this whole aspect of doing this type 1, type 2 error in little detail is to explain to you the concept of alpha error and beta error. Beta error is very, very significant in clinical studies, where if you see power of a study is very important in determining the sample size. If your power is less than 80, then the result is not considered as acceptable or good. So typically the power limit is kept as 80%. Power value, anything above 80 is acceptable. And as far as alpha level is concerned, we talk of probability value of 0.05 as the limit with respect to alpha. So this is type 1 and type 2 errors for you. Let's go now into factor interactions. We have been talking since yesterday about factor interactions. So what is this factor interaction? So basically, when the effect of factor, of a particular factor, depends on the level of another factor, the two factors are said to interact. Now let me take an example. We were discussing about baking a cake yesterday, and we discussed about the 4 factors. Now you imagine that the sugar level, which is very important for taste. Now effect of sugar level, suppose, depends on the temperature used. What I mean by that is that at lower temperature or at optimum temperature, whether the sugar level is low or high, taste is good, and obviously with the high sugar level, you are able to get the sweeter results, sweeter taste, which is expected. But when you have a temperature which is high, then what is happening probably is there is something getting charred, and your low level of sugar also doesn't give you good taste, which is ideally it is supposed to give. So your effect of sugar in the cake depends on what temperature do you use. And this is what do you mean by interaction between the two factors. So the level of temperature, so if you read this sentence again, when the level of a factor depends on the level of another factor, right? So level of a factor, that means the effect of a factor, sorry, not the level of a factor, effect of a factor, which is sugar, depends on the level of other factor, which is the temperature, oven temperature, then the two factors are said to interact. So this is how the interactions become very important for us to know. And this kind of factor interactions, you cannot get it in OFAC, and therefore we say that DOE scores over the OFAC. Okay. Now, how do we see these interactions? They are usually seen very critically, very easily, again, in the graphs. Like we see the half normal and Pareto chart. Similarly, interactions are seen also graphically. Now, what do you mean by two-way interaction? That is the second order effects on the response variable resulting from interaction of two factors, that is referred or denoted as AB, AC, or AD. Now, if I want to use again in the, explain in terms of the example of our baking of cake, then that is considered as the interaction between the temperature and sugar. So if the sugar was factor A and factor C, and temperature was factor A, then we will term it as AC interaction. So that's how you label the interaction. What do you mean by three-way interaction? Now there is third component. There were actually four factors, and the fourth factor was use of eggs, which probably gives desired fluffiness and consistency to the cake. And this also depends on the temperature that you use. In that case, what you will say that your all three factors, which is the number of eggs you use in the cake, then second is the sugar that you use in the cake, are dependent on temperature, and therefore now you will say that they are interacting three-way, or there is A, B, C. But as you would see, excuse me, there can be even A, B, C, D interaction, which means all four factors interacting. Now, you can imagine that this is a situation which is not very common. Now, even in cake example, you are very unlikely to use a temperature which will affect all the three other factors. So, in real field or reality, you will only see two-factor interaction, very rarely three-factor interaction, and very, very rarely, or you will practically eliminate the four-level interactions or four-factor interaction. Let's see that with a picture. Now, this is a very typical picture which is used in terms of some story with the office politics, and you will see that this guy ahead is trying to pull the cart, and there are four people who are supposed to help. Okay, so now what happens with this guy is he's pulling the eventual outcome is that you have to take the cart to a distance. Okay, and there is a slope which they are climbing. Now what happens with this guy? Probably he's doing helping this guy to take it ahead because there is a slope, so he's pushing it. So that is called as probably a positive interaction. What we mean here is that the two factors can interact with each other positively as well. Then this guy behind, you will see, is a negative interaction because he's pulling it behind. So it's a negative interaction. Like we saw in the example of cake, that when you have a high temperature, the taste is spoiled, which means that the temperature has a negative impact on the response, and particularly also with the sugar concentration. And if you see these two guys, there might be no interactions. Like we have seen yesterday's example, actually none of the factor interacted. So this is how you can see that there can be what kind of interaction. There can be interaction with these two guys. There is an interaction with this one and this. And now you are seeing this guy does not interact with this. Now you imagine that all of them were pushing it in one direction. Then you had a very positive 5-level interaction, which rarely happens. But this is what I was just trying to say in terms of number of factor interacting. I hope the factor interactions is clear. We move on to this graphical representation. And this graphical representation on the right, if you see the graph, wherein two lines are parallel. This is Y, which is the response, and both the lines represents the two factors. Now, when you have two factors which are having response which is parallel to each other, then these factors are said to not interact. And when you have two lines on your left of the screen, when you have two lines interacting, then what you are saying is two lines which are intersecting, then you are saying that these factors would interact. So this is how, looking at the graph, you can determine the factor interaction. So just again to recap, two factors interact if in interaction graph they exhibit intersecting or non-parallel lines. Parallel lines suggest lack of interactions, which we've seen in the paracetamol tablet. What do? We concentrate only on the main effects and two-factor interactions, as I told earlier, a three-factor and very rarely three-factor or four-factor interactions will happen. And therefore, unless you really think that these factors are likely to interact, like if you're working on a control release formulations where you are using two polymers for controlling the release, and then you are using also a binder for granulation. In such cases, it is possible that all three would kind of interact. And when you know this, and that's the, as I would say, every day, that the subject matter expert is very critical person in design of experiments. So if looking at the formulation, composition, if a subject matter expert feels that there is a possibility of three-way interaction, then that must be included in the design itself. So that's how you do. Typically, you will consider the one level or the main effect or one factor effect and two factor interactions, but not three factor. Let's go on to residuals, because I'm repeating this because we did it little hurriedly yesterday, and also we had a question there. So what are the residuals? We said that the data typically when you generate, that's your response as a part of the experiment that you do, is the signal, combination of signal and noise, whereas what you analyze as a part of DOE or design of experiments is you try to filter or you try to separate out the signal, and therefore eventually what you model is only signal. So when you subtract the predicted results from your observed results, what you get is the noise or the residuals, and this residual has to be obviously minimum so that you can see your effects very well. Now the question that I had asked was, residuals are calculated as signal to noise ratio. It has nothing to do with signal to noise ratio. Then we said adding observed value to the predicted values. Again, it's not addition. And then there is a subtraction. So that was only confusion probably, that you are subtracting the observed value from predicted or predicted from observed. So just to clarify, that the observed value are always going to be higher because it is signal plus noise. Predicted value is likely to be lower because it's the only signal. However, there are also exceptions to this because there is variations associated with the response that we generate, and therefore what is important to note is always you do observed minus predicted. So I hope that answered where we have seen all the colors in the pie chart, which is not very good. I hope that gets cleared. Now, what do you mean or what do you do when you observe such things? So we have to do something with that data when you observe. Now, when I discussed yesterday, I discussed about the megaphone effect, and I also gave example of dissolution. So the image or the graph on your left is the one which is desirable. This is what we got with paracetamol study, but the right graph is not desirable. Now, suppose you get this, what do you do? Or when do you get this? Let's do first when do you get this, and next slide we'll decide what to do. So typically what happens is when you have a response which is covered over a range, like if here you see impurity response which is ranged from 0.03% to 3.1%, which is more than 100-fold change. Or you have a dissolution value wherein you are talking of 5% dissolution and you are talking of 95% dissolution. Such a broad range of data when you have, then you are likely to see this effect. Then no significant effects found in normal, half normal plot. Now this is also very unusual because you are seeing that there is a variation in the response, but I have not seen any significant effect in the half normal plot, which means I don't see any points which are coming far away from the trend line. So again, I have a challenge there because that cannot happen. When I'm doing such a study, I should see some effect. Then we have also seen the plot of residuals where we saw a trend line and all the data was around that trend line. Now, instead of that, suppose you have S-shaped pattern, then you know that you have a challenge. And last is that when you see megaphone effect. So there are, when you have such 4 effects or 4 observation, you are likely to see non-normality. And I hope everyone remembers what is normality. Let me repeat, the normality means that your data fits into the bell-shaped curve with the, there are the criteria as we were mentioning about 67% of data residing between ±1 sigma and so on. So when you have normal data, then you can do all this statistical analysis. When you don't have your data fitting into the normality, then you have a challenge. Okay, so what do you do? There is one more help to you. I discussed with you, that there are possible observations such as megaphone effect or S-shaped curve and so on. There is also in the software provision, which is called as a box-Cox plot, which is of power transformation. So if you see box-Cox plot, it very clearly tells you the recommendation of transformation. You can see in the middle that it has said that recommended transformation none. We'll not get into details of how do we get it, how do we interpret, because it's already given. It's too much of statistics, number one, and the interpretation is already given by the software. So I think we'll not get into that. So now the box-Cox plot also have said that, okay, you need to transform the data. What do we do? No transformation means your data is as such linear. You can do logarithmic transformation, you can do square root transformation, you can do inverse transformation. Now, when we say this transformation, you really don't have to do anything. You have to give commands to the software. It will automatically do this transformation, and it will analyze. So you can do logarithmic transformation, and then you will see your data. You can do square root and again see your data. Of course, there is another check, which is the R-square regression check is also there. But it again became too statistics, so I have removed, I've not discussed, but that is also there as a check. Okay. Now, many of you who are into research would know that we do such transformation very routinely in dissolution when we are trying to determine what is the mechanism of drug release from a formulation. So we obviously do look at the linear wherein drug release is independent of concentration. Then we will say it follows the zero order kinetics. If it is logarithmic, then we'll say it is first order kinetics and so on. So it's not something which is unusual for us. But one thing very important for us to remember, whenever you do data transformation, that means your response has been transformed into any of this functionalities, then you have to use the inverse functions to when you are interpreting or when you have the model ready with you. So if you have done logarithmic transformation, then you must take anti-log when you are interpreting eventually. So this is we should not forget as an additional step. Right now, what do? We are doing all transformation. There is no ANOVA results. There's no improvement. There is nothing happening to residuals. Then we must consider some other aspects, and that other aspects could be you have to look at the design. You might have to look at the power of the study. You might have to look at particularly the noise. Do you have a significant delta by sigma, what is determined as the noise? You have a very high signal to noise ratio. So there are some of the aspects that must be considered, and then we should move on. Okay. There's one more term which is used, which is called as confounding or aliasing. Now, this is also a very commonly used term, and this is the basis for fractional factorial, and that's why we are discussing it. Aliasing is, and these terms are used many times interchangeably, though there is a minor difference, but at this point in time, it's good enough for us to understand this collectively. So when does this occur? This occurs when two factors or interaction terms are set at identical levels throughout the experimental design. We'll see this with example. Now, where does this occur? It occurs in the screening design because the design does not include all combinations of factor level. Okay, for example, if factor A is confounded with three-way interaction BCD, then what happens with this aliasing effect? So if you have factor A confounding or aliasing with BCD, then what effect you see as an outcome in this software, you cannot determine whether it is because of A or it is because of BCD. Many times you will see increased A value or increased BCD, which means that these values, effect values have been added up. Remember, the response is not added up. Response is separate. But when you do calculation in the software, which depends on what design we have selected, the response, eventually the effect which is generated for the calculation purpose is added up. If you remember, effect is determined by subtracting the high values with the high level values from the low level values, as I had explained yesterday. Again, if it is not clear, do write to us in the chat box. We'll come back to it maybe tomorrow again. You will never see any kind of aliasing structure or aliasing effect in full factorial design. Now, if you see full factorial design, all the designs are very different. And just giving you example of AB and BCD, AB and CD, because you will see somewhere they alias. This is 1 plus 1 minus 1 plus 1. Here itself you see the difference. Then rest of the design is different. If you remember, 1 means the high level, 1 is nothing but plus 1 is a high level, minus is the low level of that factor. So you will see this design is very different from this, and likewise all the designs are different from each other. So I have only shown here 12 experimental runs. Totally there are 16, but none are matching with each other. So this is what happens with full factorial. But the problem of full factorial we'll see later, why we go for fractional factorial. Now let's see fractional factorial. And I'll be also explaining you what is the meaning of 2 raised to 4 minus 1. But for the purpose of this slide, what is important for you to see this, all this AB, AB and CD, AC, BD and BC and AD. Now you will see plus 1, minus 1, minus 1, minus 1, all are same. And same happens with this. Now, when you have same, now let's understand what is the further meaning in terms of label. What it means is in a particular standard run, what you are doing here is you will be taking 30 milligram of PVP, and that is the A and B factor together and C and D factor together. Now, it's difficult for me to explain to you with the numbers, but because the factors are easily understood at the individual level and main effect. But when you combine these two, you will know this, like effect of factor A and B, which is like 30 milligram of povidone and 20 milligram of cross carmelose versus say 200 milligram of MCC, and you have colloidal aerosol, which is 5 milligram. So like that, you will see that kind of effect will be similar. I'm actually taking different factors here, and we are trying to see this. What actually it means that the not the the example, the way I told you, it is that in these two designs the labels will be same. So in AB, if PVP is at 30 milligram per tablet and cross carmelose is 20, in C and D also it will remain at the same level. Now, how does that happen? That is determined by the software, and therefore we may not get into that. But when you see these two same, you will see aliasing effect, or you cannot differentiate between the two, right? Now, similar thing you will see here in a three-way interaction. A and BCD, they are having same runs, same structure. So this will be also alias. So this is what is important for us to know, that results of these factors are going to be alias, and we cannot determine that it is because of a particular factor. And this is a big problem in our study. As I said, we have to separate out the factorial design because there is no aliasing effect. Now, towards the end of this presentation, we will also see a comparison of different fractional factorials with factorials, and you will really appreciate that time what is meant by aliasing. But at this point in time, you need to understand that the factors do alias. Let's go further. Now, there is also a term which is known as the resolution. What we mean by resolution is that experiments are expensive to conduct. Now, this is where the factorial design becomes challenging, because if you were to do 5-factor study at 2 levels, it means 32 experiments you have to conduct. And if you have 6 factors, then of course it becomes 64. Now there can be challenges of time, there can be challenges of availability of excipient, money, and so on. You can't do all these experiments, and therefore you have to cut down. And how do we cut down? We cut it down by doing fractional factorials. Okay, and we do the screening designs. Now, for the purpose of resolution, it is important for us to understand that higher the resolution of design, higher is the discriminatory power, and therefore it is preferred. Okay, this terminology is used in fractional factorial, and it is defined as degree to which the design of an experiment can differentiate between levels of interactions. Okay, we'll see again more, and that will become clear. For us right now, it is important to know that if you have a resolution 2, then main effects are aliased with one another, and therefore it is not recommended at all, which means that effects of two factor itself will appear similar. Resolution 3, main effects are aliased with two-way interactions. This is recommended only for screening because you will have two-way interactions, affecting main effects. So you will see in the example that screening designs can be used only where we have a very significant effect coming from the main effect and there are no interactions. Like we saw yesterday in paracetamol case where there were no interaction effects. Paracetamol IR, we saw the significant effect was coming from only factor B and C, which is PVP and cross Carmelos. When you have such kind of understanding, when you know such things, then you can go with resolution 3 designs. Resolution 4 will be always preferred because the main effects here, like aliasing with two-way interaction, here main effects alias with three-way interactions. And if you remember, three-way interactions are not very common. So you are kind of very confident of the main effect in the resolution 4. The two-way interactions do alias with another two-way interactions. However, again, when you're looking at main effects, I think resolution 4 design is good. And you have resolution 5 design, which is most recommended for modeling. So all these designs will become important as we see further. And this is more important certainly when the number of factors go up. Now let's see this in little more detail and with the example. So let's say we are doing some kind of lubrication study. Okay, I did take this example briefly yesterday as well. So now when we are dealing with many factors, what happens is that exploring too many factors may be really costly, as I said, and too few limits the success of your experiment. Now let's take this kind of example. Okay, impact of mixed stearate on dissolution. Now from composition, I can say concentration of mixed stearate, the specific surface area, particle size of mixed stearate itself also can impact, and there are different brands. Then process-wise, blender type, blending time, so on. So there are 4 factors here, 5 factors here, totally 9 factors that can affect the impact. Now, if I select, as you see here, too many factors, then it becomes costly. And if I select too low factor, then I cannot actually determine the impact of magnesium stearate on dissolution. So I have to strike a balance. What I do first is I will go to the subject matter expert and I'll see if I can reduce anything from this. Okay. As I was saying you or telling you that 9 factors means 512 experiments. So nobody is going to do this many number of experiments. So there is something called fractional factorial design, and they are represented by something known as a 2 raised to power 3 minus 1. All the terminologies remain same except this one terminology, which is the one additional here. This is commonly also referred as 2 raised to k minus p. 2 raised to k minus p design allows analyzing k factors with only 2 raised to k minus p experiments. What is p? P is the extent of fractionation, and we will see how that is working. The remaining thing, as I said, 2 is the number of labels, 3 is the number of factors. When you do 2 raised to 3 minus 1, it becomes 2 raised to 2, which is number of runs is 4. You can take the same thing with 2 raised to 5, and you will get 16 runs. If you have 2 raised to k minus 2, it becomes 8. Okay. Now there is one more thing. If you look at the original number 32, why this is called extent of fractionation? Because this is half of 32. So when you say P is equal to 1, it is the half fractionation. If you have P equal to 2, it is 1/4 fractionation. That's why this is called as the extent of fractionation, this P. Okay. Now we'll see, this is the common table you will see in any software where you are going to use fractional factorial. And how do we see this? You have to see first is that there is the red marks. Now these are the studies which they don't recommend. Eventually decision is ours, but they don't recommend, because you will see that these are the resolution 3 designs, and you know that in resolution 3 designs the main factors will alias with the 3-level interactions or 3-factor interactions. So this is something not recommended, but we can still do it if we want to do it. There is something which is intermediate, which is the yellow colored, where you have the resolution 4 design. So here you will see that 2 raised to power 6 minus 2, and on the subscript you will see the resolution. So here you will see the resolution 4 design, and then this is the resolution 5 or 6 designs, which is the one of the very good choice designs, which are marked as green. So they would recommend doing this design. And finally, on white, with white color, what you see is the full factorial. So you can see here, if you take 5 factors, full factorial runs are 32. And if you go 5 minus 1, then it will be 16. If you go 5 minus 2, it will be 8 factors. So this is how you read the table and you select the design here. Okay. Now we go to subject matter expert, and he says that particle size and specific surface area is going to be very similar. So please drop it. Let's not study different brands at this point in time. We can decide on the COA. So they dropped 2 factors out of it, and they've also removed 2 factors. So now we are left with 5 factors starting with 9. And now when we have 5 factors, we will have either full factorial, which is 32 design experiments, resolution 5 design, which is 16, and resolution 3 design, which is 8 runs. And we'll actually carry out these experiments, and we will compare the results to determine what happens when we use different types of designs. Okay, so this is resolution 5 design, where you will see the number of data of as we see here, just a minute, 16 experiments. You are seeing here 13 experiment runs. I didn't include the other runs because otherwise the table would become too small in font and you will not be able to see it. What is important here is the elements of the design, and then you will see the dissolution value. So this is this will follow the same pattern as we have done yesterday. When you select the design, give the factors, and automatically the the software will give you the runs, standard runs, randomized runs, and the labels of factors that you need to use for that. So everything is given for you, and you do those experiments and you feed in the dissolution data. Now, after we feed in the data, what we see first is the factorial design, half normal plot, and we see the ANOVA table. Of course, we can see Pareto, but Pareto and half normal give same results, so I've included only half normal here. As you are seeing here, the effects which are away from the red trend line is A, concentration, B is blending times, and then you see here the interaction. Okay, we are on resolution 5 design where you do see some interaction. So we are seeing CD interaction, very important. Then you have also factor C. So you are seeing that these are the important elements of your design, or the important factors which affect your response. Now, when we look at the ANOVA data, you will see here that again the concentration of mixed tier rate, blender, RPM, blending time, and CD, all effects are significant. How do we determine significant effects? Because all these values are less than 0.05, and therefore they are significant. This is the same. So the extrapolation of the results remains same irrespective of the design what you select. Okay. So we have seen these 4 effects being significant. Let's see the resolution 3. Okay, this is the design. You do the experiments and you get the data. What is it now? You are seeing now only two effects being significant. Okay, you are seeing factor A and factor D, which is blending time and concentration, are the significant impact factors, and therefore you will see that the only two factors you are seeing also in ANOVA. Okay, then we will see the full factorial design. Now in full factorial design, you will be surprised to see so many terms. You will see that concentration of mixture rate, blending time, blender RPM, then you will see CD interaction, you will see AD, you will even see ABC or ABD effects also significant. Now typically if I look at half normal plot, maybe I would have not consider AD, ABC and ABC effect. And this is the beauty of confirming your results with ANOVA table. When you see ANOVA table, the other effects are, like if you see here, BC and CBD, these effects are less than 0.05, but the level of this effect is not as high. If you see here less than 0.0001, means it has to be highly significant effect. But if you look at the AC effect or you look at the other effect, they are not very significant. So the ANOVA table tells you that they are significant, may not be the half normal plot. So as I have said that always you confirm your results with the ANOVA table. So this is the significance of ANOVA table. And now let's see what is the comparison. And that's very, very interesting. If you see full factorial, you will see that all the out of 5 factors which we were studying, only B and E did not have the effect. Now, when we did RISO 5 study, you will see that you matched with full factorial, no problem with it. But when you did fractional factorial, you did not see the effect of C. And this is little surprising because even here, fractional resolution, you are supposed to pick up main effects. And we'll see why this is not happening in short time now. Okay, so this is what we are seeing. Significant effects of A and D are captured in all 3 designs, and you have the main effects and fractional factorial RISO 5. All this exhibited similar results. Okay, now what happened to factor C? Now, if you see here, the value, p-value, which is determined, okay? Now, also we have to remember that the p-value is determined based on data that you generate. The more experiments we do, the more response that we generate and feed into the software, it will become more and more accurate. So when we have a very limited study, limited experiments done, when we did only 8 experiments, what we got was that p-value for this was more than 0.05, and therefore it became insignificant. And you will see that the p-value for C factor keeps on reducing. If you see RISO 5, it has become 0.024, which is less than 0.05, and therefore it is significant, but it's not highly significant. And now if you see full factorial, the value is 0.0001, which is highly significant. So this is the difference of the data that you get based on your input, because that is the compromise. Like, you don't want to do lot of trials, lot of experiments, then obviously there will be some compromise that you will have to do, right? So in terms of main effects, what happens, what we see is main effects, full factorial design and resolution 5's design, they are similar and they are better compared to resolution 3 design. So this is the conclusion that we are drawing for the main factor. Now let's see what happens to the two factor interactions. Now if you see 2 factor interactions, again you have these interactions like AC, AD and BD and CD. These are, you are seeing this interactions here. In fractional factorial with resolution 5, you're seeing only one interaction CD. And in fractional factorial with RISO 3, you don't even determine any interactions. Okay. So when we talk of 2-factor interaction, obviously the full factorial is much better than, or rather full factorial is better than RISO 5, which is much better than Reso 3, where you can't even determine the any interactions. So this is how the selection and the screening of the design, selection of design will happen. Now let's go to the next design, which is Plackett-Berman design. This is also a screening design. Now what happens in Plackett-Berman? So what we are saying is the fractional factorial, Plackett-Berman, and we are going to see Taguchi's design. These three are the screening designs. We have seen the fractional factorial. You can almost extrapolate the results, outcomes of the fractional factorial in terms of the interaction part with all the other designs. So we're not going to see the examples there. What we're going to see is only how the design varies. Now, in fractional factorial, the design is given by term 2 raised to power k minus p. Here, the design is given by very different things. You have to select straight away the Plackett-Berman design, and you have to give, what is the number of factors that you have. So in case of Plackett-Berman design, it is an effective design for screening a number of factors, but what at 2 levels. So you generally don't use 3 levels, and in any case, screening, you don't want to go for 3 levels. So Plackett-Berman is good for the screening. It is very useful for economically detecting large main effect, assuming all interactions are negligible. So these words are very important. You have also seen in fractional factorial that when you have large main effects, you do see them in even Reso 3 designs. But if you have the not very significant main effects, then you may miss in Reso 3. And two-level interaction will be missed in all designs, really. So screening designs are not good for that. Now in Plackett-Berman, what you see is the 4 into n experiments are needed to estimate main effects, which is 4n minus 1. So that means if you have 7, n is 2, which means you have main 7 effects, you can do it in simply 8 experiments. Okay, this is the beauty of this. So again, if you do fractional factorial with say 7 factors, it may still tell you to do, if you do 7 minus 3, 2 raised to 4, 16 trials, which is still higher compared to Plackett-Berman, suggesting 8 numbers. So this is the difference between the Plackett-Berman and the fractional factorial. Again, you are reducing the number of runs. Limitation, of course, is the low power and cannot be used for interactions. This is how the design looks like. I'm not going to get into the details of how you derive at it. If you go a little deeper into this, you can design your own Plackett-Berman 8-run or 16-run or 12-run matrix or fractional factorial design. It's not difficult, but of course, for the purpose of the webinar and looking at the time, we are not getting into this part, which is already given by the software. Now let's compare this Plackett-Berman design with the fractional full factorial. So if you have 5 factors, number of experiments will be 32. Here it is 12. This is taken from the literature. Now significant factors are B, C, D. That's the main effect. What is the level of main effect? So what it says is the high level, high B plus 1, and this is the same notations. So it is giving the similar effect as full factorial. Significant interactions BC and BCE. Of course, it is not detectable in Plackett-Berman. And there is an optimized value of Y response, which is 114 that they have got in the literature, which is 116, which is not very far. So what we're trying to say here is that if we want to leave the interaction part, we can use Plackett-Berman. But all these designs are very important only when you have lot of factors. When you have less factors, I would still recommend you to go with the full factorial designs with center points, which we'll be doing next week, in the next session tomorrow. Let's quickly go to Taguchi's orthogonal array design. And this is the most of the designs, if you see, focus on the main factors and uncontrolled factors. If you remember, I had discussed yesterday about the process part, and in process part there was noise, which had the measurable and non-measurable factors. What Taguchi's design considers is they do consider the uncontrolled variable as well. in their design. So the whole idea of Taguchi's design is that your output should be very close to your mean value, although you're allowed a kind of a leeway, with leeway in the sense for the variations. There is a range, but Taguchi's design will always focus on the center. So if you say essay, your range could be 90 to 110, but Taguchi's design say that how we can achieve always 100 as your essay. So that's the difference between Taguchi's design and all other design. There is a notation which says that Ln multiplied by S raised to factor K, which is of course the factor, and S is the number of labels. So this is very similar to what we have 2 raised to 3, or similar kind of connotations that we have for factorial design. What you have added is the Ln, which is the experimental run. So this is how we define Taguchi's design. Again, you just have to go and select in the software Taguchi's design, and this is how they suggest. And we say that 2-level factors is where you use Plackett-Berman. Taguchi's you can use for 3 levels as well. So if you have 2 factors or 2 parameters, you will use 2-level and 2 factors. L4 means 4 experimental run. So you will see that 4 and 2 levels, L8. Now this is how you are seeing, and if you see 2 raised to 7 factors, if you see, that means 2 raised to 7 will be somewhere maybe 256 or something if you go with that kind of design. Whereas in this you are able to do with only 8 experiments. That I was referring to full factorial. But even fractional factorial 2 raised to 7 minus 3 if you take, so 2 raised to 4 again 18, 16, whereas you do here in 8 design. So the difference here is the way they adjust these labels in your experimental run. So there is a lot of statistics associated with this part, how you are putting your +1 and -1 in the design, and that is where the power of determining main effects come. But these Taguchi's orthogonal array and Plackett-Berman particularly helps when you have very large number of factors to be evaluated. So to conclude, basically screening designs are used when there are many factors and which are to be screened and no interaction is anticipated between the factors. Screening designs are typically used in academia, as I was saying, where they're working on newer research areas and many unknown factors are to be examined. In pharmaceutical industry, screening design is not very common. Typically, through the knowledge, because of the generic medicine, only limited factors are required to be examined. So at the most in pharma industry, they will use fractional factorial, but more common is the factorial design with the center point, or if you see curvature effect, then the response surface methodology, what we are going to do tomorrow. So this is how you will go ahead. Screening design, which is fractional factorial, Plackett-Berman, Taguchi's orthogonal array. Then you will go to characterization, which is full factorial or high resolution, like resolution 5, 6, 7 designs. And then you will go to optimization, which is full factorial with center points, CCD or BBD. So these are the different designs. So yesterday we did the basics of full factorial with 2 raised to 3 factorial designs. Today we have done screening designs. So we'll tomorrow take up the optimization designs with full factorial with center point, CCD and BCD. So this was the last slide, and thank you so much. Today we finished almost on time, so that's great as well. Right, we are open for questions.
Yeah, thank you for the explanation of experiment. We'll now questions is from Swarup Ansare, and we have a 3-factor interaction of %ratio of polymers if combination is used, and the level of plasticizer while performing DOE study for coating of tablets.
Okay, so can we have a three-factor interaction of percentage coat, ratio of polymers, if combination use, and a level of plasticizer will performing of? Yes, of course, you see, you can have these three-level interaction. Because eventually what is your response, you have to see in terms of response as well. Your response is going to be say a dissolution or the tablet appearance, or you're going to looking for some of the tablet effect, a coating effect, like orange peel or something. What is your response? It depends. But if you're looking at coating efficiency, then both the polymers and plasticizer are likely to interact. Likely, I said, of course, you have to prove with the design. And yes, you should be using a higher resolution design or a full factorial design in such case. Right. Can we take next question? Okay. I think there is some connectivity issue with Dr. Anand, but I can take one more question, which is from Prashant Chaturvedi, who says that how to decide that which transformation method has to be used if the data is non-linear. Okay, this is good question. So there's no real kind of I should say a guideline or anything. This is little trial and error. You see the data, then you will see go with logarithmic transformation. Now, typically, I give you one example of impurity level where it is 0.03 to 3.1. So this appears that there is an exponential increase, and in such cases, log transformation does help. So what people typically do is first transformation they will do is the log transformation, followed by, if that doesn't work, then they will go for other transformation of square root or inverse kind of transformation. Okay, right. Then there is another question is okay. Okay, there is again Prashant Chaturvedi has asked another question. Could you please elaborate more on how to decide level of resolution and extent of fraction? So basically, this will be decided based on what you are looking at. So as I told, that if you are very sure of the main effects and significant main effects, then you were likely to select a relatively lower resolution design. And of course, when you select a relatively lower design, you will also get the accordingly fractionation that will come with it. So that itself will determine how many runs you want to use. So and obviously you will know, or you will anticipate that there are no interactions. So if such things are there, then you will select a particular design. I have already given the recommendations in one of the slides, that when you are looking at a very significant main effects, then you will go with RISO 4. And if you are likely to see less significant effect as well, which does of course impact the resolution response, then you should prefer response rather resolution 4. Also, one very important thing that determines this is that what is the flexibility in terms of number of experiments that you can do, you have. Now, if you have excipients which are readily available, you are not using very expensive excipients, and you don't have to do really very difficult experimentation in terms of making tablets or so on, then I would always prefer you going with RISO 4 or higher version of resolution, because it will give you a lot of confidence in the data. So, cutting down on number of experiments just to reduce work, I think should not be the objective. You have to look at all the aspects together at one go, and then you take a call. Yes, I can see Dr. Anand back.
Yes, sir. The next question is by Banu Buddha. What is the main determination of resolution? Yes.
I think this was the same question. I just answered the same question, which was asked by Prashant.
Okay. So we'll take up the next question. Sir,
Yeah.
could you please
Okay. Could you
elaborate
please
more
elaborate?
than how to
Go
decide
ahead.
the level of resolution?
No, again, this is the same similar question. Can we go for
Okay.
next?
We'll take the next question by PharmaViz. DX has significantly higher runs for same problem in comparison to JMP and more complex to use. I request to the speaker to detail the pros of DX with respect to JMP.
Uh, we are not really comparing here any software. The aim is really to discuss about the basic part. And personally, I have also not compared the two softwares. Uh, so I will not be able to give any answers why Design Expert has significantly higher runs compared to JMP. I probably believe that you might be referring to little different designs because these terminologies are pretty same. If you say factorial design, then 2 raised to 3 is common across, so you can't have different designs. So if you are talking about in one placket Berman and in other Taguchi, then obviously the number of runs can differ.
Okay, we'll take up the last question for today because of scarcity of time. This question is by Mrs. Deepa Warrior. Uh, Sir, I want to know when we have 4 responses, and out of 4 responses, if we get curvature for 2 responses insignificant, then do we have to repeat all the experiment runs again?
Uh, I think, Dr. Anand, you'll have to take one more question because this question will reserve for tomorrow. We are going to study the curvature effect tomorrow. So let's not preempt that. So as a last question, since we skipped this, you can take one more.
Okay. So the next question is by Karishma Mul Ranjani. Is it better to directly select factors and levels based on literature, or is it better to go for screening?
Uh, okay, there are multiple answers to this. First answer is that ideally all the design of experiment data or literature that you do, they suggest that you do prior some experimentation and you will have knowledge of that understanding of your process or your composition, formulation, and so on. And then you go for actually design of experiment or any particular design. So basically, you do carry out some basic experimentation and then you go for the DOE-based experimentation. Second thing is, of course, literature will help you in this. And again, we are referring to, probably the academic space where we are dealing with a lot of unknowns. If we talk of industry, as I mentioned earlier, most of the things, effect of factors are known because we use standard excipients. The terminologies are grades are standard. We know all that thing, so it becomes very easy to select in pharmaceutical industry. But if you talk of academia where we are doing novel research, probably you want to see the literature and understand that if you have taken a particular composition, you are developing liposomes, you are developing nanoparticles, then what are the likely factors, likely process factors, composition factors, which will impact your response. So you will do that literature, or you will talk to somebody, subject matter expert like your guide or your senior, and take their help to decide this. And eventually, like as I gave you also example that we had 9 factors, but we could reduce them to 5 after talking to an expert. And those 5 factors, again, you can't do factorial, full factorial. So you still have to do screening design. So what it means is that looking at the problem at your hand, you will have to go through this step by step and determine what is the best solution. you may straight away go with the full factorial design, or you may decide to do screening, or you may decide to do a literature search and then follow a different path. So anything is possible. Whatever is your problem, that has to be looked into detail to decide the next course.
Thank you, sir. Thank you for your patient answering for all the questions. Uh, so we conclude for the today, and one important announcement that the link for e-assessment is given in the description box of the YouTube video right now. So do join us for tomorrow session, that is day 3 at 6 p.m. Till then, stay safe.
Thank you.
Thank you.